orang berkunjung ke pasar untuk nak tengok ikan apa nak beli. So, hari ni kita akan beli ikan crispy. So, datang.
Datang. Right, so nanti bila you guys pilih ikan, boleh buka dia punya sirip atas. Okay, contohnya macam ni kan, nampak kan dia ada yang hitam-hitam tu. Itu dia punya ni lah, cacing. Okay. Right, lepas tu satu lagi cuba buka mulut dia kalau ada.
Dan kalau store owner benarkan ya, jangan suka-suka hati buka.
Make sure lepas buka tu beli. Alright. Lepas tu satu lagi boleh check dekat operculum dia. Kalau ada cacing lah.
Kadang kat operculum tu akan ada crustacea, alright, ataupun kutu ikan.
Alright. Satu ni kita ambil.
Dr. Hafiz, berapa saiz kita nak ambil?
Kita cari yang apa ni? Lebih kurang
16 cm kan?
15 cm kat atas lah.
Ataupun dalam saiz palma lah. Tapi palm pastikan besar ya, jangan kecil lah.
Sekejap. Tadi ikannya ada, mungkin ada dia punya apa? Cuba buka mulut dia kalau ada yang cymotoid isopod. Aduh. Ni tak ada. Tak apa.
Okey, lagi satu untuk pastikan ikan tu segar, pastikan mata dia ceras, bukannya macam merah membara. So this is quite fresh, it is good. You want something which is fresh supaya parasite tu pun dapat macam hidup. Kalau dia terlalu lama dan terlalu busuk, parasite pun sepatutnya bergantung pada host dia. So kalau dia terlalu merah, ini is not a good sign also. You also want to look at the sisir. Pastikan sisir dia cantik, bersinar. There's a little bit of like a mucous membrane. So that is where most of your external parasites, when you're going to do a slime, what do you call this? Apa ya Dr. Hafiz?
Smear.
Sure, yes. Bila kita nak buat smear nanti, so pastikan sisir tu tak keluar. Alright? So you want a nice big fish like this.
Alright. Juga nanti kita akan bawa ke lab, kita akan check lagi lebih detail. Okay. So, tadi saya katakan dah cara-cara ikan harga dah nanti dah jenis ikan mana yang dipilih, saiz apa yang dipilih dan sebagainya. Dan kita pun dah remind ya untuk you pilih dengan kebenaran fishmonger ataupun penjual ikan. Alright. So the next step ialah kita untuk identify ikan apa yang telah kita beli, kerana you have to be reminded that dalam dalam macam dekat pasar tu lah, sometimes dia bercampur antara spesies-spesies ikan, kerisi dan sebagainya. Of course lah, kerisi pun ada banyak jenis. So dalam section pertama ni, Dr. Mel dan saya akan cuba terangkan macam mana untuk identify spesimen ikan kerisi yang you guys bawa balik berdasarkan buku Fishes of Terengganu kan?
Ya, betul.
Fishes of Terengganu. Right, Mel, you nak terangkan sikit?
Yes, saya nak terangkan. Alright, so if you're looking under Fishes of Terengganu, check out the book here. So under page 127 to 131 is all about the fish that you're dealing with, which is called MMB3353, or the common name is threadfin breams or monocle bream. So you're going to see a few different species. So we won't go into a lot of details about identification. However, what are the important characters that you need to identify is when you're looking at your fish, the first thing you need to do is make sure the fins of the dorsal fin and the soft ray fin, alright, the spiny dorsal fin and the soft ray fin are opened up. So how are you going to open it up is by using a small pin. You can even use a scarf pin to do a small insertion at the flap here, alright, so that it opens up and spreads up. The dorsal fin are very important aspects of identification for your fish. Also, another important aspect is the caudal fin or the tail fin. So you want to ensure that it's not broken up. It's all very nice and clear. And another aspect that you want to look at is the body. So make sure the scales are all nice and shiny. if your fish tends to get a bit too dry, always make sure it's moisturized, okay? And so these 3 keys are very important for identification. So in the case for our species, how we've managed to identify it to Nemepterus furcosus is because the soft ray fin has a small yellow band right at the tip of the soft ray, of the dorsal soft ray fin, and the caudal fin does not have any yellow bandings like some of the other species. Also, with the body, there's actually no banding whatsoever if you were to see in the other species within the book. So that is how we've managed to come to the conclusion of Nemepterus furcosus for our species. Now, in your case, other than identification from the book, please take a picture of the fish that you have purchased from the market and ensure that you have identified it according to this book. Also verify back with both Dr. Hafiz and myself if you have identified it correctly according to the book. Alright? So pretty much that's it for this part.
Identification process. So basically in this book there's only about 10 species of Nemepterus. So you guys just have to cross check with the specimen that you have which one is yours, and just confirm it with us if you hesitate. So, right?
Alright. So see us and stay tuned for the next part of this lab, which is the dissection of the fish. Thank you.
Thank you. Alright, so sekarang kita akan move kepada cara untuk dissect spesimen yang kita dah dapat tadi. Dan untuk dissection ikan ni terbahagi kepada 2 jenis lah. Satu ialah untuk pemeriksaan ataupun examination bahagian luaran ikan ataupun external examination, dan yang bahagian kedua ialah internal examination ataupun pemeriksaan bahagian dalaman ikan. Right. Bagi pemeriksaan luaran ataupun external examination, organ-organ yang kita akan periksa ialah seperti mata, dorsal fin, caudal fin, sisik dan operculum dan sebagainya, dan juga bahagian mulut. Dan bagi internal, kita akan buat insertion ataupun potongan daripada kloaka hingga ke bahagian mulut ikan, dan kita akan periksa organ-organ dalaman yang kita keluarkan selepas tu. Right? So mari kita tengok ikan bahagian yang kita akan periksa bahagi external examination. Okay, untuk external examination kita akan semak ataupun periksa of course lah, macam saya kata tadi, mata. Jika ada sebarang jangkitan pada mata, kita akan nampak lah seperti cacing ataupun cyst dan sebagainya di bahagian mata. Alright. Kemudian kita akan cuba buka bahagian sirip ni. Okay, contohnya dekat sini kita boleh nampak kita boleh nampak yang terdapatnya cacing. Mungkin Dr. Min boleh tunjuk. Okay, cacing di bahagian yang melekat di antara rays ataupun spiny dorsal fin lah kan. Right, dan kemudian kita check juga pada bahagian caudal fin jika terdapat sebarang jangkitan lah. Okay, last kali kita akan cuba buka bahagian operculum. Okay, ini adalah operculum ikan, bahagian bawah ikan. Kita akan buka operculum tersebut dan kita akan cuba kita akan semak lah ataupun periksa di sini. Kadang jika terdapat infection ataupun jangkitan cacing, kita akan dapat kita akan nampak dia akan melekat di bahagian pipi ikan ni. Okay, kiri dan kanan lah. So you boleh check kedua-dua bahagian tersebut lah. Right.
I would also like to add yang kalau what other kind of parasite yang you will get. So selain daripada cacing, sebenarnya itu ikan, crustacean paling senang nak nampak kat bahagian operculum, bahagian operculum dekat sini, ataupun kalau mulut dibuka. Alright, so you have to check. walaupun kutu ikan tu terdapat internally, it's considered as an external crustacean. So sekiranya ada crustacean yang masuk dalam digestive organ, it's more because it's an accidental ingestion. So sepatutnya semua crustacean terdapat secara luaran. Di dalam mata sebolehnya kita nak kena korekkan dengan kita punya forceps and check kat dalam mata tu sendiri sekiranya ada apa-apa cacing yang terdapat di bahagian mata. So selain tu, pastikan anda belahkan sirip untuk tengok sekiranya ada cacing ataupun copy pot ke apa yang terlekat di bahagian sirip di belakang, sebab mereka suka berteduh di bahagian sirip ke sini. And terkadang dalam bahagian sirip ni, selain daripada cacing yang kita jumpai tadi, kita mungkin akan mencari bentuk cyst. Cyst ni nampak macam buih-buih kecil yang terdapat pada bahagian sirip dan juga pada bahagian sirip caudal fin juga. Dan juga kita nak tengok pada sirip di bawah sini, kadang kita akan ada nematode, copy pot yang mungkin akan terlekat dalam bahagian sirip. So sekiranya kalau kita nak buat smear, kita akan ambil forceps, alright. Pastikan ikan kamu tidak kering. So pastikan dissection ini dibuat secepat yang mungkin dalam keadaan di mana dia ada slime ataupun mucous membrane, supaya kita boleh smearkan and spear ikut bentuk arah sirip dia. Alright. So when you when you smearkan, so pastikan you ada slide. Alright. I know dalam keadaan di rumah agak susah untuk dapatkan slide, tapi mungkin
you boleh skip lah.
Yeah, so you boleh skip. Tapi kalau you're still interested to try the technique, kanak-kanak semua boleh letakkan dia punya sisik di atas glass slide, and then you put it on your glass slide with a bit of the slide. Alright. So and then you put penutup slide atas slide itu sendiri untuk tengok di bawah.
Alright. So sekarang ni seperti mana yang saya cakap tadi ataupun yang kita bagi tahu tadi, pada ikan ni terdampatnya cacing di bahagian sirip, di bahagian sirip dorsal ni. So apa yang you guys boleh buat ialah ambil daripada dissecting set ataupun dissecting kit you, pointed needle, ataupun kalau tak ada pointed needle, you boleh gunakan ni lah, macam ni, kita kata needle yang tajam ni ujung dia, dan you pierce through rays ni. So bagi dia koyak lah, right? Koyakkan. Okay. You boleh koyakkan rays tersebut. Okay. Dan tarik dia ke atas, bagi dia koyak. Okay. So that sekarang ni cacing tadi tu dah expose pada udara lah, right? So you boleh nampakkan dia masih hidup. Okay, dia boleh hidup. So you guna the pointed needle untuk tarik dia keluar. Slowly jangan bagi dia putus. Try as gentle as possible lah. Okay, nampak ini cacing yang kita dah korek keluar daripada sirip tadi. So you letak dalam piring lah. Kalau apa-apa piring yang ada kat rumah, letak sedikit dengan saline water. Okay, macam ni. Right. So you boleh nampak cacing tu masih bergerak lah. Right. So this is nematode. Okay, nematode group yang biasanya kita jumpai di bahagian sirip ikan berisi, di bahagian operculum, dan some species macam anisakis dan sebagainya kita akan jumpa di bahagian internal, okay, dalam perut. Yang tu nanti kita akan dissect lah daripada sini. Alright. So habis ke section ni?
For this section, we've done Salam kanak-kanak parasites. So welcome to the fourth part, And the last part of our lab for today, which is the dissection of fish to expose the internal parasites that we have. So Dr. Hafiz is going to be assisting me to help dissect the fish. So Rafi, what you're going to do this, and you have to be very careful of how you do this, very patiently and slowly, is that you're going to be running your utensils, particularly the knife, through the cloaca or pretty much the anus of the fish, right up to the operculum, and then you're going to lift the flap gently by running the scissors through the abdominal section of the body in order to expose the organs. So later Dr. Hafiz will explain to you what are the organs, what are the internal organs of the fish, and what likely parasites that you're going to get from this dissection. Okay, so let's move right in. Okay, so before we start, you need to have a scalpel, a scissors, and a forceps. Now, if you don't have a scalpel, what you can use is you can use a sharp knife. And then if you don't have a dissecting scissors, you can use a scissors. And if you don't have a forceps, you can either get one in a $2 shop, or you can use a tweezer that you can use to pluck the nose hair or your armpit hair. Alright, so let's dive right into the dissection. So Dr. Hafiz now is actually going to start with the scissors and the is it the sharp end or the blunt end? So Dr. Hafiz is going to sharp with the blunt end. So you're starting right at where the anus is. So you have to make an insertion. Alright, and then you lift. If you notice, he's lifted the skin up. Why you have to lift the skin up is that you do not end up cutting the internal organs of the fish. So lift the skin up and then cut along the fish right up to the operculum.
So just make sure the reason why we put the blunt end inside is to make sure that it doesn't tear off or pierce the other organs inside the fish body, right?
So as Dr. Hafiz is cutting along, notice how he's also cutting the pelvic fins of the fish. So run the scissors along that pelvic fin. It's going to be a little bit hard, but do it very gently and very slowly. Okay. Alright. So once you fish nearby the operculum, so you go upwards to where the fin is. Alright. And if you notice, there is actually a lateral line that runs along the fish. So that lateral line, some fishes actually have a scute. This one doesn't, but it's very apparent. So you run that scissors along the lateral line of the fish. So again, like before, slowly lift up the skin so that you won't end up cutting the organs. So what Dr. Hafiz is doing is advisable, where you hold the skin in one hand and also cut along as you're going. So now Dr. Hafiz has exposed that skin and the fins, and what you should be able to have and see is the internal organs of the fish.
So this is actually a female specimen because they have these eggs gonad here. So that's how we know it's female or male. So you can record that in your data sheet. So the individuals that you are examining is whether female or male, right? So you are already exposed to the internal organ. So this is the gut or the stomach, and this is the liver. And if you can take out all of this organ, okay, you can see the other part, which is the here is the intestine here, and underneath here, underneath this this is the swim bladder, and underneath the swim bladder you can find this the kidney running along the vertebrae of the fish, right? So you can take out this.
Gently take it out.
So I have I will make a cut here. Okay. So now you have the this the stomach, and then you have this the intestine. That. So you can examine this under the microscope, or you can use your magnifying glass to check if there's any, for example, nematode on this organ, okay, the internal organ. So right now, if you notice, there's a lots of nematode on top of this organ. Okay, you can already see, for example, with your naked eyes here. Okay, let me try to show it to you. Here is nematode, the one that already terkeluar bila you dissect tadi. Here. And then here, ni pun ada juga. Okay, nampak tak? This is also nematode here. And then a lot here also. You can see this here, a clump of nematode. What's it? Okay, tak ada banyak.
Wow, that's a lot.
Yeah. So this is just to remind you that it's very important whenever you are, trying to cook fish, or you're going to make fish dishes, just to make sure that you clear this part,
The internal organs as thoroughly as possible.
As thoroughly as possible. Right. So if I were to move on to this internal organ here, you can also see that there's a lot of nematode is actually attached to this organ. Okay, for example, here there's nematode. Banyak lagi ya. Kat sini pun ada, dekat on the liver. Nampak tak? Okay. Dekat liver. Tu. Ni.
So you will notice also that some of these nematodes are still trying to struggle to attach onto the organs, where some came off very loosely. So for those that are trying to attach very hard, we would suggest that you slowly tweeze it a little bit, but not to aggressively pluck it off, otherwise it can detach. So while Dr. Hafiz is doing this, please be reminded to take note of where you were actually finding these nematodes. If you were finding, say, 5 on the liver, so make sure on the data sheet that we have provided for all of you that you write down 5 nematodes from that particular organ. And then say if you find 5 other nematodes from the stomach, you should write as such.
Alright. So then what we can do is we can try to separate this organ. Okay, now I would like to separate this intestine from the gut or from the stomach and put in one of the other petri dish. Okay. Okay. So like this, just put it inside. Okay. Still you can see there's a nematode here. Okay. Okay, here, these are all nematode. But what I want to do now is actually to kita nak scrap off isi daripada intestine tu, dan kita akan cuba tengok ada tak diarrhea ataupun cesto dalam perut, dalam intestine ikan keris ini. Okay, sementara itu kita tarik dulu mana-mana yang kita jumpa.
I would also suggest that while you're getting very engrossed with finding so many kinds of parasites in your internal organs, please do not forget to at least make sure that your fish is still moist and not left exposed, in order for whatever parasites that are remaining to not actually dry out.
Right, so now saya akan tarik keluar isi perut daripada intestine ni. So what you can do is actually to grip, okay, dia menggunakan forceps at one end dan tarik intestine tersebut lah. So in this way, you akan tolak keluar ke semua isi perut daripada intestine tadi. Okay, so bila isi perut dah keluar, that's how you can take a look at what's inside this intestine lah. Okay. So now maybe I need some water. For this one, maybe we will need this microscope lah. Check. Okay, kadang-kadang bila you tengok dalam air macam ni, saline water, if you left it for a while, you akan nampak kalau ada cacing, dia akan start untuk wiggling lah, dia akan start bergerak dan sebagainya. Then that's how you can pick up these animals. Okay, sekarang ni saya tak nampak sangat, so nanti saya akan tengok dengan lebih detail lah bawah mikroskop. Okay, so the same process will be repeated for this for the stomach. Okay, you boleh buka dan you boleh check dalam you boleh check lah dalam perut tersebut apa yang cacing yang ada. Okay.
Alright, so we've come towards the end of our dissection. I hope you had lots of fun. So don't forget, as much as you're having fun, to keep with the hygiene and protocols of our work at home. And let's, for this bonus round, why not we give you an insight into how these parasites look like. Stay tuned. So this is sort of a sneak preview into what we actually have from this dissection. So we don't have a lot of parasites, the diversity, we have a lot of nematodes here. So we're going to have a look at how the nematodes look like, since we do understand that you don't have a microscope at home. So what are you likely to see under a microscope? So have a look here. So basically what you're seeing here is a nematode. So nematodes, in comparison to other worms like say flatworms are literally flat, as roundworms they actually somewhat like cylindrical and worm-like. Okay, so the other thing is also they're not segmented as what you would find in a flatworm. So in here, what you can see towards the bottom left, it's esophagus, it's its mouth part, and then it tapers down to its tail towards the end. Now, with identification of species, it is important to look at certain key features like say the esophagus, the head, the heart, and even the tail. Alright. So now in this case, with this microscope and with this magnification, it's not easy to readily identify based on the species, but with the aid of more advanced microscope or even like SEM, it can actually help taper down to what species we're looking at. But this is just a generalization of what you actually are seeing. So this is coming close towards an adult phase, but also in our sections here, you will see that this is another nematode which is not fully developed as this one, and some you would actually find in a cyst, or like a coating around this, as like such. Okay, so this is still a stage, is still underdeveloped, and something like this. So they'll be very much coiled. So which is important that when you are actually about to eat any of the fishes, we would advise that you actually remove all of the internal organs, which also includes the gills. Alright, so you ought to wash it thoroughly, and if you can rub a bit of salt on it, it's actually quite helpful. And then obviously it's best to cook your fish so that you don't encounter these guys in your system. So now the case with little regards to the nematodes, some nematodes are not zoonotic, meaning to say that they are they can use for zoonotic parasites, it means that they can use humans as a definitive host. So sometimes it's accidental, but some cases are such that it can use you as an actual host. And that's why you have the whole anisakibase issue with sushi incidents and stuff. So that is the case for the nematodes. Any take, Dr. Hafiz?
I think that's pretty much what we wanted to show you today. So of course, daripada sampel yang kita dapat ni, there are a few, I think the nematode itself is a few species among them. And to identify them according to species, we need to do a more thorough checkup lah, which means that we need more advanced equipments, more advanced analysis and so on, right? So for this introductory exercise, I think this is already good for you to gain the exposure for this parasite examination. Okay, just to be reminded that you can also find another sort of type of parasite from your specimen, not necessarily nematode, but maybe you will find more interesting parasites from your specimen. For example, the isopod, maybe you will find more of the the sea lice and so on at your particular location. Okay, so that's all from us. I hope you will enjoy this video and you can follow as well as can perform the examination yourself at home.
Take care, bye-bye.
Thank you guys, goodbye.